Hello there. My name is Peter Barbuti. I am an associate research scientist at Columbia University in New York. I'd like to thank the organizers today, especially Philip Fortier, for giving me the opportunity to talk at such an important event. The title of my talk is The Role of Lipids in Alpha-synucleinopathies. These two words, lipids and alpha-synucleinopathies, is a bit of a mouthful and needs a little bit of introduction. So I will do this during the course of the presentation, and by the end of the presentation, hopefully you will come to understand the importance, or sort of equal the importance of lipids to alpha-synucleinopathies, like I do. And also at the end of this presentation, I'm going to share with you, on behalf of my co-authors, actually, some new research that we're working on here at Columbia. So what are lipids? So simply put, lipids are a variety of molecules that do not dissolve in water. For instance, in this picture on the left, you see olive oil being poured into a glass. So clearly, oil and water do not mix. We know this. And there are also a different variety of lipids. We have fats, wax, grease, oil. They're all different types of lipids. And in fact, you get different types of oil. Olive oil and castor oil are different types of oil. So clearly there are a variety of lipids, and different lipids clearly have different functions. Now, when we think about the brain, apart from water, the majority of the brain is actually lipids. Actually, after fat cells, adipose cells, the brain is the second fattiest tissue in the body. So now we have established there are lipids in the brain, the next question we have is, what are they doing and why are they there? So let's try to break this down. The brain is made up of cells. So each cell has a barrier separating it from other cells. And around the cell is a barrier. This barrier is called a cell membrane, or specifically speaking, this cell membrane we refer to as a phospholipid bilayer. So in this rather complicated image here, we see this phospholipid bilayer is simply a cell membrane, which separates the external fluid, so the out of the membrane, compared to cytoplasm, which is the inner membrane. So as you may imagine, this barrier serves to sort of keep the structure of these the cells, but also allow transport of material, substances inside and outside the membrane. Now, within this barrier, we have this phospholipid bilayer, because it contains two layers. We have so we have this phosphate head group, this little circle, and then tail, which is a fatty acid tail. So these phosphate head groups can be all different. So we can have different lipid groups. So there's a lot of complexity within this lipid bilayer, and each of these different lipids can have also different functions and are responsible for different signaling patterns as well. Also within this lipid bilayer, we have a substance called cholesterol, which I'm sure you're all aware of. Now, cholesterol is very important for the membrane because it aids the fluidity of the membrane as well. In addition, the inner and outer leaflet of the membrane is also quite different. We have different phospholipids which populate the membrane compared to the inner leaflet and the outer leaflet. And this can be very important as well, which can relate the function within the cell and compared to other cells. And just to add an extra bit of complexity to something which is slightly complicated, is that within these little tails, these sort of fatty acid tails, we have an additional differences. We have these fatty acids can be saturated fatty acids, like, for instance, butter is saturated fatty acids, or they can be polyunsaturated fatty acids, like olive oil, for instance. They are clearly different. Now, in the same way, these saturated fatty acids are a carbon-carbon tail, and they are very stiff. Whereas polyunsaturated fatty acids contain one to many different kinks in this membrane. So many double bonds relate to alterations of the structure and the composition of this membrane allows it more fluid, more permeable, so different substances can pass through. Okay. So now we've introduced lipids. Let's see what's going on in the healthy brain. Well, I'm sure you're all aware that in the brain we have many different cell types, but I guess the most well-established are neurons. Well, neurons transmit information by electrical or chemical signals within the brain, from the brain to the spinal cord and throughout the body. And also within the brain, we have other cell types. We have helper cells. And I guess the most important of these helper cells are oligodendrocytes. This is particularly important for MSA, multiple system atrophy. So oligodendrocytes are responsible for forming a myelin sheath. Now, a myelin sheath is it surrounds the neuron, and it's important for neural transmission. This myelin sheath wraps around the neuron like an insulator, like an insulating wire in your household when you do the electrics. There's no electrical wire exists on its own. It's surrounded by this insulating layer, which it needs for function. So of this myelin is approximately over 70% of lipids, and within those lipids, approximately 40% of that is cholesterol. Now, if you think relating to other cell types, that would be maybe 25% cholesterol. So clearly the fluidity, cholesterol is very important in the myelination of the neuron. So the neuron is surrounded by lipids, which is incredibly important for its function. Now, in addition to these oligodendrocyte helper cells, there are also cells, astrocytes. Now, astrocytes are other helper cells. They provide metabolic and nutritional support to the neurons. And also we have another helper cell type called microglia. The microglia you can consider as the brain's immune system, which helps to sort of prune synapses in a way that a gardener would prune the hedges in his or her garden. So everything is organized and ordered and neat and tidy, and in a way that the neuron connections mirror that. If the connections are uncontrolled, then clearly there's a problem. So the microglia has an important functional role as well. So what happens in MSA? Now, MSA is not a uniform disease. There's MSAP, which is more linked to Parkinsonism. So it has more Parkinson's features, such as rigidity, and patients would be also more bradykinetic, slowness of movement and speed, for instance. Whereas MSAC would typically affect the cerebella, into cerebral ataxia, which would be difficulty coordinating, difficulty walking, speech, hand and eye movement. Now, what goes on in MSA to these cell types, the neurons and the helper cells? So within the neurons exists normally is this protein called alpha-synuclein. Now, somewhere during the course of this disease, this protein begins to aggregate. And also during this aggregation process, this aggregated alpha-synuclein transfers from this neuron to these oligodendrocyte helper cells. This is referred to as a prion-like transfer. So this is transfer of an aggregated protein from an affected area to an unaffected area, which I'm sure we'll hear about in many of the other talks. Now, what happens once this occurs is that these aggregated alpha-synuclein in these oligodendrocytes, which refer to as glial cytoplasmic inclusions, they clearly affect the oligodendrocytes and affect its demyelination it affects the myelination of the neuron. Fundamentally, what it means is that the neurons become demyelinated. The electrical insulation wire is gone. So these neurons are also very vulnerable. In addition to this, we have sort of proliferation of these other helper cells, the astrocytes and microglia, which also linked to the disease as well. Fundamentally, this accumulates in the degeneration of the neuron. So what is particularly important is that within the disease, as MSA progresses, the cytoplasmic inclusions, these aggregates in these oligodendrocytes, correspond and correlate to the severity of the neuronal loss. So clearly, this is a very important thing, these aggregated alpha-synuclein. So I've mentioned alpha-synuclein. What is it? So it's a protein. It's a small protein, highly expressed in neurons in the brain, but it's also highly expressed in other cell types like the colon, the lung, and in red blood cells. Now, in different parts of the body, it has specialized functions, and alpha-synuclein is also the key protein of Parkinson's disease. And there have been many decades of work on alpha-synuclein, and despite knowing a little bit more about the protein and what it actually does, the fundamental answer is that we still don't really understand. We don't really know. Alpha-synuclein as a function of this protein is still incompletely understood even now. But what we do know is that we know the structure of the protein. It has 3 domains. It has an N-terminus, which binds lipids. It has a NAC domain, which is the part of the protein which begins to aggregate. And there's a C-terminus, which is the part of the protein that binds calcium. Again, further linking alpha-synuclein to lipids is that alpha-synuclein is a cholesterol binding partner. So how does aggregation process occur? Like I said earlier, we don't really know. But what we do know is that alpha-synuclein exists quite happily and functionally. It functions as a monomer. It functions as this native form here. Now, somewhere along this process, it begins to misfold or folds incorrectly, and this is linked to the disease process. Now, this misfolded protein can take on different types of forms, different types of structures, which people refer to as oligomers and fibrils, but fundamentally, it ends up clumped and aggregated. Well, also, this means is that it's no longer a functional protein. And we do find the aggregates of the alpha-synuclein in the post-mortem brain samples as well. So we clearly know that this protein is pivotal in these alpha-synucleinopathies as well. So I mentioned alpha-synucleinopathies, so let's try to reinforce this in a little bit more of detail. So in this slide, you can see we have these 3 circles: Parkinson's disease, dementia of Lewy bodies, and MSA. And these are, generally speaking, the 3 main alpha-synucleinopathies. And these different size of circles also correspond to the relative numbers of patients affected with these different diseases. Now, as I mentioned, in MSA, you can have different regions of the brain affected. The MSA-P affects the striatum, MSA-C affects the cerebellum. Yet what is unique amongst those, what is common with MSA is that the oligodendrocytes are affected. And what we found is that we have aggregated alpha-synuclein is in these oligodendrocytes. Now, when we look at Parkinson's disease, the cell types affected are dopaminergic neurons in the substantia nigra, a different region of the brain. And also, when you look at dementia of Lewy bodies, we have a different region of the brain affected as well, which is the cortex, but again, and a different cell type. So not just dopaminergic neurons, or per se, multiple neuronal types are affected in dementia of Lewy bodies. Yet what is consistent between these different cells, the oligodendrocytes, dopaminergic neuron, and other neurons in general, what's common is the aggregated alpha-synuclein. Now, I've shown these in different colors of green, orange, and blue to represent the different structures, the different strains of this protein that scientists find, pathologists find in this disease. Yet fundamentally, it is the same protein. It's the same protein, but a different structure, a different strain. Leads to different diseases in different cell types. Yet it's still alpha-synuclein. So even though it's very complicated, I hope I've sort of elucidated or tried to give a little bit of an insight that clearly it's the same protein that goes wrong. So if we try to bring this back to looking at lipids to MSA, so researchers have tried to find genes and mutations that cause MSA, yet currently there isn't really a consensus. Yet genes can have variations which are normal, totally normal. However, some of these variations are associated with increased risk of MSA. So interesting variations that alter lipid metabolism are the genes that encode, for instance, the gene SNCA encodes alpha-synuclein. The gene GBA1 encodes glucocerebrosidase, which leads to important protein in Gaucher's disease. Gaucher's is a lipid storage disease. MAPT encodes tau. Tau is a protein that is synonymous with Alzheimer's and frontotemporal dementia. Now, interestingly, in MSA, this gene CoQ2 encodes coenzyme Q10. And I'd like to spend a moment to just briefly discuss CoQ10, because population studies in East Asia, particularly in East Asia, have found that there's been a reduction in coenzyme Q10 in MSA patients. So let's ask to think, well, what does this coenzyme Q10 do? So coenzyme Q10 protects lipids from lipid peroxides. So if you think of peroxides, you go to the hair salon, peroxides used to bleach your hair. So your hair goes from brown to blonde, for instance. This dyeing process, this bleaching, this is a stress of the hair. It leads to oxidative stress. The same process happens within the cell to these lipids. These lipids become stress. So, and also what we realize as well that with lipids and MSA is that we have higher level of serum cholesterol, again, tying cholesterol and alpha-synuclein to MSA. So higher level of cholesterol associated with a decreased risk of MSA, which incidentally ties together with coenzyme Q10 as cholesterol regulates CoQ10. So this is sort of the background of what we know up until now. So what did we do at Columbia? So in our study, we looked at, we were fortunate to have a section of brains which were donated to Columbia University of people who had multiple system atrophy, MSAP, and Parkinson's disease, and also healthy control individuals who were of similar ages. So we looked at the affected region in these two different diseases, which are of course linked by alpha-synuclein. So we looked at the striatum, part of the region of the brain region affected in MSAP, and the substantia nigra, part of the brain affected in Parkinson's disease. And we looked at the lipid profile. So this slide is a busy slide, shows the lipid changes we observed in MSA patients. So firstly, we looked at the striatum of the MSA patients against healthy individuals. On the corresponding axis, we have what we found as well were significantly altered lipids from the MSA patients to the healthy controls. We saw alterations in cholesterol esters, alterations in ceramides, and alterations in some of these phospholipids that regulate the cell membrane. Also within this vertical axis, within these lipid groups, we have correspond to different structure and different lipid chain length. So as you can see, from 16 to 18 to 22, this refers to the increased number of carbons. And then after the semicolon, from 0, 1, 2, this refers to the amount of double bonds within the carbon. So how polyunsaturated that there are. So fundamentally what we can see is that we see significantly altered lipids in the post-mortem striatum of MSA patients compared to controls. Now, what we were able to, so the next thing we did as well was to compare these lipid alterations in the MSA post-mortem samples to the affected region in Parkinson's disease, which is the substantia nigra. So interestingly, we found the same lipid groups were altered. So we found the cholesterol esters were altered, yet the individual lipids identified from this lipidomic profiling were in fact different. The next thing we wanted to do was we looked at the PD postmortem brains, and we decided to look at different regions of the brain which correspond to different susceptibility of the disease. So the substantia nigra has more neuronal cell loss than the VTA, with the hypothalamus and the substantia innominata, different regions of the PD brain, less affected. And we can see that within Parkinson's disease, particularly, we have sort of greater disease. We have different lipid alterations compared to different disease severity. I also apologize, I failed to mention that the red refers to the fold increase in lipid changes, and the blue is the fold decrease in lipid changes. But what we saw is that compared to MSA, different lipids were found in the individual MSA brains compared to the PD brains. Again, what we found as well, that broadly speaking, the magnitude of the lipid alterations were less in MSA than in PD. However, certain lipid changes were more pronounced in MSA. And what was quite interesting for us was this particular lipid, phosphatidylserine. So we really wanted to sort of explore this a little bit further and try to investigate the mechanisms underpinning these changes which we've observed in the postmortem brain samples. So to do this, we try to model the disease in a dish. So we're fortunate enough to take we generated stem cells from healthy individuals, and we generated stem cells from an individual who had a multiplication in the gene that generates alpha-synuclein. It was a Parkinson's disease patient, so this patient had too much alpha-synuclein, but nonetheless, it's a model of this synucleinopathies. So this individual had more alpha-synuclein. So we were intrigued to see what is the effect of alpha-synuclein here. So we generated neurons. So obviously, skin can only make skin. From the skin cells, we made stem cells. From the stem cells within the dish, we generated these dopamine cells, which are more affected in Parkinson's disease. We also used gene editing to remove the alpha-synuclein gene. So what we were left with was a sort of dosage effect. We had healthy cells with a normal level of alpha-synuclein, and we had unhealthy cells with a toxic level of alpha-synuclein, and then also cells with no alpha-synuclein at all. What we were able to find, we did the same lipidomic profile as we did in the postmortem brain samples. And fundamentally, what we were able to find is that the patient neurons replicated what was very consistent with the postmortem brain studies. So we can find here, each of these colors correspond to these different lipid groups identified. And these 3 bars correspond to these different cell types, which refer to the last slide of the cells without any alpha-synuclein, with a healthy level, and with too much alpha-synuclein. So clearly showing that alpha-synuclein alters lipids. Now, we were particularly, so we looked at phospholipids, and we looked at this lipid, phosphatidylserine. We're particularly interested in this lipid, as we know from many studies that when this lipid, this phospholipid, which is typically found on the inner leaflet of the plasma membrane, when it's externalized, is the signal for cells of the immune system, such as microglia or other cells, to attack the neuron itself. So it may lead to an underlying, it may lead us more clues in the mechanism involved, or why these loss of cells are occurring, why is this degeneration occurring. So we were interested to find that the alpha-synuclein dosage a mirror image between the cells with the duplication, the cells that are too much alpha-synuclein compared to the cells that are no synuclein at all, which led us to think that perhaps these lipid alterations are associated with alpha-synuclein dosage. So the next thing we wanted to do was, let's investigate this. So where is this phosphatidylserine regulated? So to do this, we looked in our neurons, the patient-derived neurons, and within the neurons, we have specialized compartments. The ER, which in the endoplasmic reticulum, that generates proteins, and the mitochondria, which is the powerhouse of the cell, which generates ATP, which generates energy. So between these two domains, there's this specialized domain called the MAM, mitochondrial associated ER membrane. And the MAM is responsible, it's a specialized domain, responsible for many different things. However, one of those things it is responsible for is the lipid homeostasis within the cell, and it is at the MAM domain that phosphatidylserine, this particular lipid, can be generated. So we performed in our study a series of experiments trying to tease together how to identify the mechanism of what really occurs. So we again looked at these cells with different levels of synuclein dosage, from no synuclein to a healthy level to too much toxic synuclein, and we're able to see that there's a dose-dependent increase in this particular phospholipid, phosphatidylserine, with alpha-synuclein level as well. So we wanted to investigate perhaps we can identify the mechanism involved. So you may not be surprised to know that everything is a little bit more complicated than it seems, and this is exactly the same as well. So there are two ways that we can generate this lipid phosphatidylserine, which is in blue. One is via one enzyme, PSS1, phosphatidylserine 1. The other is PSS2, phosphatidylserine 2. A long story short, our series of experiments, we were able to find that when there is increased level of alpha-synuclein, it begins to turn off this PSS2 enzyme, and the activity of PSS1 drives out of control in this, or maybe drives out of control. It sort of upregulated significantly, which leads to this increased level of these phosphatidylserine, which we see in these cells with too much alpha-synuclein. So I know I think it's a good time to stop. So let's summarize what we know. So we know that there's compelling evidence to suggest that lipid metabolism, dysfunctional lipid metabolism, is shared across the entire synucleinopathies. We perform lipidomic profiling of the postmortem brain in regions, affected regions of MSA, the striatum, affected regions of Parkinson's disease, the substantia nigra, and we can show that, yes, there are similarities, the same lipid groups are identified, but fundamentally, there are different specific lipid changes. And also, the magnitude of those changes are fundamentally less in MSA than PD. Now, clearly, a little bit more research needs to be done to hone in on the subtypes of these lipid changes and the specificity of them. But clearly, this is very interesting and very exciting data. Now, when we look in the patient-derived cells to be able to replicate these findings of the post-mortem PD brains when we look at synuclein dosage, we can also see that there's an interplay between alpha-synuclein and this particular phospholipid phosphatidylserine, and in such that increased alpha-synuclein increases this level of phosphatidylserine that I previously said is associated with neuroinflammation and attack of the cells by the immune system. So all in all, linking the lipids and alpha-synucleinopathies, but also neurodegeneration and inflammation. So I think with that in mind, I'd like to thank you for listening. I'd like to acknowledge the funders and also for the many people who contributed to this work. And thank you very much.